Good morning children, I am Isis. Now I am going to teach you Pleiotropy, an important phenomenon in genetics, Pleiotropy. Pleiotropy means the phenomenon in which a gene can control more than one phenotypic character. Normally a gene or a pair of alleles can determine or control only a single character, but in Pleiotropy, one gene means a pair of alleles. For example, capital T can control only one character. Whether it is homozygous or heterozygous, it can control only one character. So this gene means a pair of alleles. A pair of alleles together form a gene that control a single character normally. But in Pleiotropy, a gene or a pair of alleles that together form a gene which can control more than one phenotypic character. Let's see an example. Okay. So example is, so we can write single gene can control more than one phenotypic trait. Phenotypic trait. Trait means character. Okay. So example, inheritance of starch grains or starch synthesis. Starch synthesis we can write. Inheritance of starch synthesis and seed shape in pea plants. In pea plants or in pea seeds. Okay. So the inheritance of starch synthesis and seed shape in pea plants or in pea seeds is an example for Pleiotropy. Let's see in detail. So, in pea plants or in pea seeds, the starch is synthesized very effectively by the homozygote. So, very effective starch synthesis is done by capital B homozygote. Okay, so the starch grain size is large. So large grain size. Large starch grain size. Okay, and less efficiency in starch synthesis is shown by small p, that is also homozygote. So the starch grain size is small sized grains. Okay, the grain size is small because this gene show only less efficiency in starch synthesis. And another gene, heterozygote, that show intermediate sized grains. Neither large nor small. An intermediate, a medium sized grains are produced by this gene. Is it clear? So once more we can see, in pea seeds, the gene which is having a homozygote, capital B, they are very efficient in starch synthesis, so they are producing large sized starch grains. But the gene which is having the homozygote small b, they are very less efficiency they have, so they are having small sized starch grains. And the gene which is having a heterozygote, capital B, small b, they are producing intermediate sized starch grains. Medium sized grains are produced by this heterozygote. Is it clear? And after maturation, the seeds are showing their exact shapes. When we consider the shape, this capital B show a round shape. They are round shaped seeds. Okay. And the small b gene show a wrinkled shape. And this heterozygote show a round shape. That means they resemble one of the parents. That is, here we can see this is the dominant character. Am I right? And when we consider the starch grain size or the grain size, this is an incomplete dominance. Am I right? When we consider the size of the grain, this is an incomplete dominant. Why? This is the dominant character. Large size is the dominant character. That's why we are giving capital letter. So this offspring or this genes does not show the dominant character such as large size. It is medium size only. So there is an incompletion of the dominant character. That's why it is an example for incomplete dominance. Okay. And when we consider the shape, here we are considering the shape. The capital B is dominant and small b is recessive. Am I right? Capital B is dominant and small b is recessive. Okay. Here capital B, small b is incomplete dominance. So we can say this capital B or this gene can control more than one character, or this gene, the gene which has two alleles. Which are the two alleles? Capital B, small b. And these capital B and small b can control more than one phenotypic character. Which are the phenotypic characters? Size of the grains and shape of the seed. Both are controlled by the same gene. And this phenomenon is called Pleiotropy. Or we can say more than one character is influenced by a single gene. Okay, so the single gene, suppose this may be a single gene. If we are taking this as a single gene, this can control the size of the grain as well as the shape of the seed. If we are considering this small b, that can control. If we are considering the small b as a single gene, it can control the size of the grain as well as the shape of the seed. Okay. And if we are taking capital B, small b as an example, this gene can control the size as well as the shape. Is it clear? So one gene can control, one gene means a pair of alleles. A pair of alleles together form a gene which can control only one character at a time, normal cases. But in Pleiotropy, it's a peculiar phenomenon in which a gene or a pair of alleles together form a gene. That gene can control more than one phenotypic character. Is it clear about Pleiotropy? Okay. Now we can do an interpretation. That is, we can write genotype, then phenotype. Genotype and the phenotype. We can put it in a column. Okay. And here, in this phenotype, we have to see the size of the grain and shape also. Size of grain or size of seed and shape. First, capital B is the genotype. What may be the size then? Size is large. Am I right? What is the shape? Round. Okay. And in second case, small b, small sized grains or small sized seeds, and wrinkled shape. And the heterozygote, capital B, small b, the size is intermediate or medium. And the shape is round. Is it clear? So we can say one gene can control more than one character. And here, this, when we consider the size of the grain, this capital B, small b show incomplete dominance. When we consider the shape of the seed, this capital B, small b show dominant character. Or we can say capital B is the dominant and small b is the recessive when we consider the shape of the seed. Is it clear about Pleiotropy? Okay. Next we can see the inheritance of two genes or dihybrid cross by Grigor, Johann, Mendel. Next we can see inheritance of two genes. In this dihybrid cross, Mendel was taken two characters and the characters were seed color and seed shape. Let's see the seed color and seed shape. So here we can see the round yellow seed with the cross with the wrinkled green. So we can see the cross here. The phenotype is round yellow and it is crossed with a wrinkled green. This is the phenotype. And the genotype, capital R, capital Y, capital Y. Why these are given capital letters? Because in the first generation, in the F1 generation, they are showing a round yellow. So the offsprings were round yellow. So we can conclude that the round yellow is the dominant character. That's why he was given capital letters, capital R and capital Y to this round yellow character. Okay. So here capital R, capital Y, small r, small y we can give for wrinkled green. Here the law of independent assortment is applied. I told you the law of independent assortment is based on dihybrid cross. So here at the time of gamete formation, the separation of this pair is independent of this one. That means when two pairs of characters or tribes are brought together, these are the two pairs of characters. This is the shape and this is the color. When two pairs of characters are brought together, the segregation or separation of one pair is independent of the other. So, the gamete is capital R, capital Y. Is it clear? So, this is the law of independent assortment. Here also, the gamete is small r, small y. This is the gamete. So, here there is a cross between these two. So, normally the dominate over the recessive, and the next character also dominate over the recessive. And the F1 generation is round yellow. So this is the F1 hybrid or F1 generation. And after that he was conducted self-pollination of this F1. So we can write when F1 self means self-pollinate. Okay. So here the same parent, which is the parent, the phenotype of the parent is round yellow and round yellow itself. Because we were, Mendel was taking this round yellow for self-pollination. So tell me the genotype, capital R, small r, capital Y, small y. The same genotype, capital R, small r, capital Y, small y. Okay, here also we can see the law of independent assortment. So there are possibilities of gametes. Which are the possible gametes? Capital R, capital Y. Here, capital R, small y, small r, capital Y, and small r, small y. The same gamete is produced by the second panel, capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. So clear? Normally we are doing, so taking this gamete and cross with these 4 gametes. Another second gamete is taking and cross with the 4 gametes, but this is not possible here. So we can use a column that is called a Punnett square. Punnett square is used to do the crossing of this dihybrid cross. Let's see the Punnett square, which consists of 4 into 4, 16 columns. Okay, so this side we can write the genotype of the female. Here we can write the genotype of male. Okay, so these are the male gametes and this is the male gamete and these are the female gametes. Here, this gamete need to cross with the 4 gametes. Okay, so let's see. Capital R, capital R, capital Y, capital Y. Then this need to cross with this one. Capital R, capital R, capital Y, small y. So like that we have to fill this column. Okay. Now the crossing is done in the Punnett square. You can see. So in this cross, in the dihybrid cross at 2 generation, we got round yellow, round green, wrinkled yellow and wrinkled grey. Here the round yellow is the parental character. Am I right? And wrinkled green also parental character. Am I right? So here the wrinkled yellow and round green, these are the recombinant characters. Is it clear? So here the F2 progeny show parental characters as well as a combination of the parental character, and that is called recombinant. So which are the recombinant characters here? Wrinkled yellow and round green. So these are the recombinant characters. Let's see what is the phenotype, phenotypic ratio and genotypic ratio. Okay, dihybrid phenotypic ratio. Phenotypic ratio, for that you have to find out the expressive character. How many round yellow, how many round green, how many wrinkled yellow and how many wrinkled green. You have to find out. So here we can see 9 round yellow, right? You can count it, and 3 round green, 3 round green, and 3 wrinkled yellow and 1 wrinkled green. So we can write over here 9:3:3:1 is the dihybrid phenotypic ratio, and the dihybrid genotypic ratio, you have to find out how many homozygous round yellow, how many heterozygous round yellow, then how many homozygous wrinkled green, and how many heterozygous wrinkled green. Like that you have to find out. Anyway, you will get the genotypic ratio 1:2:4:2:1:2:1:1. This is the dihybrid genotypic ratio. Is it clear? You can do it individually. And that's all about the dihybrid cross. So in dihybrid cross, we can see the F1 progeny resembles the parental character, one of the parental characters, that is the round yellow, when it's self-pollinated, the F2 progeny shows parental character. Along with the parental character, they are showing a combination, and that is called a recombinant character. Recombinant character. Recombinant means a combination of both the parents. That is called a recombinant character. Which are the recombinant characters here? Round green and wrinkled yellow. Is it clear? Now we can see the next topic, chromosomal theory of inheritance. And this chromosomal theory of inheritance was discovered or proposed by Walter Sutton and Theodore Boveri. These two scientists proposed this chromosomal theory of inheritance. According to the chromosomal theory of inheritance, the chromosomes and genes are showing parallelism in the behavior. So the theory postulates are, first one, both the chromosomes and genes occur in pairs. Okay, so Theodore Boveri and Walter Sutton observed a parallelism in the behavior of both chromosomes and genes. And their findings were both chromosomes and genes occur in pairs. And at the time of gamete formation, they separate or segregate. And these paired gamete restored at the time of fertilization. Okay, so the second point is segregate during gamete formation and restored during fertilization. Okay, actually these genes are located on chromosomes. And they are showing parallelism in the behavior. Let's see the points. So there is a comparison between the behavior of chromosomes and genes. So the first point, the chromosomes occur in pairs, genes also occur in pairs. And they segregate during gamete formation, only one gene pair. That means only one of each pair is transmitted to a gamete. And in gene also, the segregate during gamete formation, only one of each pair is transmitted to a gamete. And the third point is, independent pairs segregate independently of each other. And one pair segregates independently of another pair. Both the statements are same. So is it clear about chromosomal theory of inheritance by Walter Sutton and Theodore Boveri? Hope you understood everything about dihybrid cross, chromosomal theory of inheritance and pleiotropy. That's all for today.